Welcome to MRU E-Learning, powered by Malla Reddy University, where science meets curiosity and learning comes alive. Myself, Mrs. Darshini Mahesh Pujar, Assistant Professor, School of Agricultural Sciences, Malla Reddy University. In today's session, we'll explore the fascinating world beyond the boundaries of Mendel's garden piece into the world where inheritance is always as simple as dominant and recessive. One of the most fascinating chapters in genetics, which is Post-Mendelian inheritance. We all know him, that is Gregor Mendel, who is regarded as the father of genetics for his groundbreaking experiment on garden pig. By analyzing the inheritance of contrasting traits, Mendel formulated the law of segregation and the law of independent assortment. So, which explain how heritability factors are transmitted from parents to offspring. His findings established the foundation of classical genetics, emphasizing discrete inheritance and statistical predictability of trait expression. However, subsequent research in earlier 20th century by several geneticists revealed that many traits in plants, animals, and humans do not conform strictly to Mendelian ratio, among which Carl Correns, Hugo de Veres, and Teschmark, who independently rediscovered Mendel's work around 19,000, who were among the first to observe deviations such as incomplete dominance in Mirabilis jalapa. Later, like William Bateson and Punnett, who identified epistasis and gene interaction in case of poultry and sweet peas. While Thomas Hunt Morgan and his group demonstrated linkage and crossing over in case of Drosophila melanogaster, establishing the chromosomal theory of inheritance. So all these discoveries collectively explained Mendel's principles, forming the foundation of Post-Mendelian genetics. So all these concepts which integrates both classical and molecular understandings of heredity. So now let's decode how these modern genetics reveals the expression of techniques that make biology even more exciting by looking into the major types of post-Mendelian inheritance. The first one is incomplete dominance. So the next, codominance, multiple alleles, lethal genes, and then the epistasis gene interaction, polygenic inheritance, pleiotropy, sex-linked inheritance, and environmental effects. And the last one is maternal effects. So these types together explain how inheritance pattern go beyond basic laws. So we shall start. Mendel always observed complete dominance of one allele over the other for all the 7 characters which he studied in garden pig. So example, round was completely dominant over wrinkled, and for seed shape, yellow for in case of cotyledons, and for the other flowers like even in case of pod and stem traits. But it was observed that neither allele is completely dominant over the other, resulting in heterozygous phenotypes, that is intermediate between 2 homozygous form, which is called as incomplete dominance. So this phenomenon was first observed in case of Mirabilis jalapa, commonly known as 4 o'clock plant. So now in case of 4 o'clock plant, so there are 2 types of flowers. One is red and the other one is the white flower. So when these white and red flowers are crossed, plants produce plants in an intermediate flower, so which is pink, which will be produced in F1 generation. When this F1 progeny was selfed, so we will be getting Punnett square, we will be writing a Punnett square in such a way, so where F1 produces gametes in this form. So where it produces homozygous dominant flower color and 2 heterozygous pink flower and a recessive white flower which is produced, where we get both phenotypic and genotypic ratio in 1:2:1 ratio. But in case of Mendelian genetics, we get 3:1 as phenotypic ratio and 1:2:1 as genotypic ratio. A perfect example of how genes blend their expression. So next condition in which both alleles of a gene in heterozygous individuals are fully expressed, resulting in phenotype that simultaneously display the characters of both alleles, which is called as codominance. Unlike incomplete dominance, where traits blend, but here codominance, both parental traits appear side by side. So example, a very good example is coat color in case of cattle. So now what happens when a white cow is crossed with the red cow? The offspring shows both red and white color, so producing a Ron pattern. So in codominance, both alleles contribute equally to the phenotype without one masking the other. Further, Mendel always observed two allelic forms of a gene. So in such a way, so he have got two different forms of gene. But now cases are known where a gene has more than two allelic forms, where existence of more than two alternative forms of gene within a population controlling a single trait, so which is known as multiple alleles. A perfect example of multiple alleles is ABO blood group in case of humans and also coat color in case of rabbit. So the first example, the ABO blood group, which is controlled by a single gene I, and it is present on the chromosome number 9. So this gene exists in three different forms, that is IA, IB and I. So when we combine these genes, we get four blood groups, that is IA, the dominant one, which is group A, IA, IB or heterozygous, which is group B, and one more is IA and IB, which is group AB, which is considered as a universal recipient. So this shows both multiple allelism and codominance, as IA and IB are equally expressed in both the blood group. And the last one, which is recessive one with the recessive trait group O, which is universal donor. So this is about ABO blood group. So another example is in case of coat color of rabbits. It is controlled by single gene with four alleles. First example is in case of agouti. So this has full color and is known as wild type, which is represented by dominant C with a brown fur color. And the next one is chinchilla. So this is lighter than agouti. This color is dominant over Himalaya and albino, the other two, which has black tipped with white fur. This is represented by CCH. And the next case is Himalayan. The main body is white, while the tips are and ears are and the snout are colored. So this color is dominant over albino. So this is represented by CH. And the last is the rabbit type, which is albino, which is completely pure white and which is recessive, which is represented by a recessive C letter. So both examples show how one gene can exist in several forms, creating genetic diversity. So the next concept is where Mendel always observed independent assortment of genes, so in this particular form. But there is a phenomenon in which two or more genes located close together on the same chromosome tend to be inherited together during meiosis, so which is called as linkage. The concept of linkage was first discovered by Thomas Hunt Morgan while studying the fruit flies. He noticed that certain traits like body color, wing type did not follow Mendel's law of independent assortment. Instead, they were inherited together provided the genes on the same chromosomes are linked. So these are the genes which are inherited together and produces parental types. Other ones are those which undergo recombination. Linkage is one of the foundation of modern genetics and also to study the chromosome mapping. So coming to the next type of post-Mendelian inheritance, Mendel always found that findings were always on equal survival of all the genotypes. But there are conditions where some genes when present in certain combination causes death of an organism, so which is called as lethal genes. In the presence of such lethal genes, the normal segregation ratio, which should be 3:1, is modified into 2:1 ratio. So lethal genes can be dominant and recessive depending on how they express their effect. So let me explain you with an example. The French genetist Cugnot discovered a lethal gene affecting coat color in case of mice. He found that yellow coat color in case of mice was produced by a dominant gene capital Y. while its recessive gene Y determines the normal black or grey coat color. Further, he found that all the mice with yellow coat color were always heterozygous, and he was unable to find a mouse which is homozygous for Y gene. So what he did, he conducted an experiment by crossing a yellow coat color mice with a black coat color mice. So we'll be mentioning the gametes in such a way. He observed yellow coat color mice and a black coat color mice in F1 generation. When these F1, so which is yellow coat color mice, which is heterozygous, were selfed for F2 generation. So he selfed and he got such type of progenies. He got 4 different progenies with 2 yellow coat color mice, which is heterozygous, and 1 black coat color mice, which is homozygous, and the 1 coat mice, which was dead. So where instead of 3:1 ratio due to lethality, both phenotypic and genotypic ratio were 2:1. So this happens when homozygous yellow condition is lethal and such embryos do not survive. On the other hand, recessive lethal genes causes death only when present in homozygous condition. In case of rice, the genes responsible for green pigmentation is essential for photosynthesis. When a homozygous green plants are crossed with the heterozygous green plant, it produces a progeny which produces a homozygous green plant and a heterozygous plant. Then these heterozygous plants are selfed. So it produces 4 different types of progeny. A homozygous green plant, 2 heterozygous green plants, and an albino plant which obtained in homozygous recessive condition. When the gene is homozygous recessive, the seedlings lack chlorophyll and appear white or albino, finally causing lethality. Similarly, due to lethality, both phenotypic and genotypic ratio is 2:1. So thus, lethal genes, whether dominant or recessive, plays a crucial role in controlling the population balance and maintaining genetic stability. So the next concept, so Mendel always observed 9:3:3:1 ratio in F2 for dihybrid cross. But there are cases where 2 or more genes influence a single trait and the expression of one gene depends or modifies the action of other. So this phenomenon is called as gene interaction. Some of the important types of gene interactions are complementary gene action with the ratio of 9:7, where purple flower color is sweet pea depends on the both dominant gene whenever both of them are expressed. Otherwise, the flower color will be white. So one more is the duplicate dominant epistasis with a ratio of 15:1 with an example of OND and ONless rise. And then inhibitory gene action with the ratio of 13:3. An example of this type of gene interaction is found in case of anthocyanin pigmentation in case of rice. Then supplementary gene action with an example of grain color in maize with red, purple and yellow kernels with a ratio of 9:3:4. Then comes the polymeric gene action with the ratio of 9:6:1 in case of barley, which affects the length of awns. And the last concept is about dominant epistasis with the ratio of 12:3:1. An example of dominant epistasis is found in case of fruit color with the white, yellow and green. So all these gene interactions explain why real life inheritance is more complex than the Mendel's ratio. In the next condition of post-Mendelian inheritance, sometimes a single gene influences more than one trait in an organism. So this interesting phenomenon is called as pleiotropy. A classical example of pleiotropy was observed in case of white eye allele in case of Drosophila. So a white eye allele. This allele affects eye color. So for example, in which is white. And then shape of spermatica, organs which consist of capsule and a tubular stalk. The capsule is the main sperm storage area. And then the fecundity and testicular membrane. In case of Drosophila, fecundity represents the reproductive output of female, while the testicular membrane plays a critical role in male fertility for sperm development. So it reminds us that genes often play multiple roles in the growth and function of an organism. So in another case where Mendel always observed that each character is governed by single gene, but Later, Nelson Allais observed that some characters are even controlled by several genes, and each of such genes have additive effect in the expression of character. So this concept led to the foundation of polygenic inheritance. So that is inheritance of quantitative trait that is controlled by two or more genes, each having a small and additive effect on a phenotype, which is called as polygenic inheritance. In contrast to pleiotropy, where one gene affects many traits, polygenic inheritance is just the opposite, where many genes together control a single trait. So some of the examples are like human color, kernel color in case of maize, and even grain yield in case of crops. So let me explain you with one of the example of human skin color. So when a very light skin color woman marry a very dark colored man, so what happens? We'll be getting a progeny which is very dark progeny is obtained, which is controlled by 3 different genes. So, and each dominant allele contribute to the amount of melanin pigment produced. When these very dark progeny was self to produce F2, it produces wide range of skin tones rather than just 2 or 3 different types. So next time, when you see the diversity in human skin color, remember it's not one gene at work, but a symphony of many genes blending their effects. So that's the beauty of polygenic inheritance. So another example is about multiple factor hypothesis, which is found out by Nelson Allais, with 16 progenies of 1:4:6:4:1, which produces dark red, red medium, light red and white kernel color. So these examples explain why traits like skin color in humans, kernel color in maize, and even the yield are not simply dominant or recessive, but show a range of variation. Okay. So next, coming to the next concept. Have you ever noticed why same plant grows differently in 2 places, or how color of some animals changes with season? So this happens because genes does not work alone. Their expression is influenced by environment. So the environmental effect refers to the influence of external factors like temperature, light, and even the nutrition and chemicals on the expression of genes. Every trait we see in an organism is a result of both genotype and environment. In a simpler terms, genes provide potential, but the environment decide how that potential is expressed. As I said, even if 2 organisms have the same genes, different in environment can lead to different phenotype. So which is because of the result of genotype and environment. Okay, some of the examples are hydrangea flower, Himalayan rabbit, and also yielding crops. Even the nutrition acts for the development of, especially in case of Drosophila. So as I said, let me explain you with an example. So the color of hydrangea flower depends on the soil pH. Acidic with low soil pH produces blue color flower, while the alkaline soil with high pH produces pink color flower. So this is because of aluminum availability in high pH soil condition. So, okay. The other example is fur color in case of Himalayan rabbit, which contributes by a gene that's sensitive to temperature. The pigment producing enzyme works only at cooler body parts like ear, nose, pouch, resulting in black tips and a white body. So that's the magic of nature. The same gene can paint different pictures at different environments. So remember, genes determined with an organism can become, but the environment decides what it will become. Okay. Here's the last concept. Which is, did you know all the genes are found in, not all the genes are found in nucleus. Some traits are inherited to the cytoplasm also. So which is called as cytoplasmic inheritance. So the phenomenon where the phenotype of offspring is determined by the genotype of mother rather than the offspring's own genotype, which is called as cytoplasmic inheritance. Other features of cytoplasmic inheritance are actually they do not follow Mendelian inheritance. They have reciprocal differences, and also the traits which are expressed are sure to produce metabolism, because since mitochondria and chloroplast are involved in energy and pigment formation. Some of the classical examples of cytoplasmic inheritance, let me explain you all. So one by one. So one of the best known example of cytoplasmic inheritance comes from a freshwater snail. So these snails show two types of shell coiling. One is right-handed or dextral coiling, which is clockwise, and one more is left-handed or sinistral coiling, which is anti-clockwise. The coiling direction of offspring does not depend on its own genotype, but on the genotype of mother. So if the mother genotype is dextral coiling, all her offsprings will be dextral. And in the same way, if the mother's genotype is sinistral, all her offsprings will be sinistral. So this is about the snail example. So other example is the leaf color of 4-o'clock plant. So here it depends on the type of egg cell, not because of the pollen. If the egg cell comes from the green branch, all the offsprings will be green. If it comes from a white branch, all the offsprings will be white. So similar example like izope trait in case of maize, male sterility in different crops. So next time you study inheritance, remember not all genetic information is in the nucleus. Some comes from cytoplasmic world that powers every living cell. So all these were the different cases of post-Mendelian inheritance. Yes, and that's how genetics evolved from Mendel's garden play to the complex world of post-Mendelian and inheritance. So that explains not just inheritance, but the true diversity of life. Thank you.